My guest today, Dr. Joachim Frank, is a German-American biophysicist at Columbia University and a Nobel laureate. He is regarded as the founder of single-particle cryo-electron microscopy. That's a technique which today allows us to see biomolecules with the resolution of an atom. He shared the Nobel Prize in Chemistry in 2017 for his contribution in developing single-particle cryo-electron microscopy. He has worked at places like University of Freiburg, München, TU München, Max Planck Society, JPL, Caltech, Cornell University, UC Berkeley, University of Cambridge, New York State University, and Columbia University. He has won a bunch of other awards and prizes throughout his long career. Dr. Joachim Frank is also a writer, photographer, and the father. He is a truly inspiring character, and it was a lot of fun to talk to him in English and in German. Maybe let's talk about how your inventions have partially contributed to combating pandemics.
Yeah, it's very interesting. 2012, I told you, was a great invention. An electronic camera was came on the market that is able to record single electrons, and so my technique all of a sudden became ripe for atomic resolution. So since 2012, there have been many high resolution structures, and coincidentally, this was really also a time when large pandemics came up. I'm talking about MERS and Zika, dengue, Ebola, and then finally the COVID.
Corona, yeah.
Right. So I just wanted to emphasize that in all these cases, CryoEM, single particle CryoEM, has made very decisive contributions to the development of drugs, development of vaccines, and development of effective antibodies.
Mm-hmm. And so could you specify how exactly? So basically by looking at the atomic resolution of the virus itself, you can construct proteins and antibodies that directly combat the virus. Is that correct?
Yes. You can, for instance, you can see where antibodies bind. You can have viruses that are incubated with antibodies. You can image them, and then you can see exactly where antibodies bind. Mm-hmm. And so you can get an idea of where are the effective epitopes, the places that are recognized by antibodies, mm-hmm. and how to develop antibodies that are more effective in a particular case. Now, it's very interesting what happened in the case of the COVID-19. You have heard about the mRNA viruses, mRNA vaccination. This is a completely new technology, and what is being done is the particular structure which constitutes the line of attack of the virus, the spike protein, you have heard of that, can simply be created synthetically. And so one can expose the body to this spike protein, which of course is not infective because it doesn't have the virus behind it. So using mRNA, messenger RNA, you can make the host, namely us, make this spike protein, which is a totally hostile protein, which is immediately recognized by the body as something foreign.
Exactly.
And it makes antibodies against it. Now, here, Cryo-EM has been instrumental in making this technique effective, because the spike protein as such has a part that is very flexible, and only in a particular position of that flexible domain can it actually be recognized by an antibody, or in such a position it is essentially recognizable as something that could possibly invade the body. Now, people have developed a particular version of this which doesn't have this flexibility, but instead it is fixed in one position in which it is always recognized. Okay. And so Cryo-EM has been used to ensure that we have the spike protein, the synthetic spike protein, fixed in this position.
Okay, so without the cryo-ER, they would have been in the dark. Yeah. So cryo-EM, they would have been in the dark about trying to put together a protein that may or may not be. So basically, where cryo-EM was crucial is to look at the actual product of the research and confirm, okay, that is our spike protein.
Yeah, that's the spike protein that we want. It has the features that we want. Okay.
So I understand that it's very crucial to be able to see it. From what I know, it usually is just enough to have our antibodies recognize the protein. So we do not necessarily have to see it with our eyes, so to say. It is usually enough to just have it recognized by our antibodies. So why was it so crucial that we could actually see it?
Well, you just make sure that the ideas that you have put into this research actually work.
Mm-hmm. So you verify basically that,
you can verify
what you wanted to make is what you made.
Right, and in this case, a mutation was used, a mutation that is at a crucial place that makes sure that a certain domain stays in a fixed position rather than being flexible and all over the place.
Okay, so what you mean is that actually in the mRNA vaccine, you do not get the original spike protein. The spike protein from the virus was kind of like engineered in a way that it's now fixed, and it's easier for the body to build antibodies against it, right?
The mRNA contains instructions on how to make this spike protein. And so the engineering of this mRNA was a major feat of
never seen before in vaccination. Yeah, I mean, I think the only time they use that a bit maybe is in to combat cancer, if I'm not mistaken.
No, I don't think so. Not with mRNA.
That's where I heard was first deployed.
Oh, okay.
And so okay.
And then another way of how Cryo-EM is used is to see where exactly antibodies bind.
So you can see the two molecules coming together and actually clicking, so to say, basically, right?
Right.
What's the time resolution of Cryo-EM?
The time resolution. You can the normal kind of Cryo-EM. It takes at least a few seconds to get it all done.
So it's not like a movie. It's not like a
it's not film. Like a movie. Now I happen to work in time-resolved Cryo-EM, and there one can really work in milliseconds. And the way that works is there's a microfluidic chip, and the two components that you want to react and follow the reaction are put into the two input channels. And then you can stop the reaction in the microfluidic chip at any time, and then spray the product onto the grid. So you can get time resolutions of something like 20 milliseconds, 50 milliseconds or so.
That's basically what a movie is, right? It's about 30, 40 milliseconds.
Yeah. Right.
Wow, exactly. So one thing I want to ask is also how this is a team effort. So CryoEM itself, and I think the entire project of CryoEM today would not be possible with only one person. Could you confirm that? And could you say something about like how many people are needed to develop something like that?
Oh, to develop it. If we talk about the development, that has gone on over decades now and has involved many groups, many teams that worked on the various parts of the technology, like how to prepare a sample, the development of the instrument itself, and then the data processing. So there were specialists that worked on all the different aspects of
Thousands of people, probably, right?
Yeah, probably hundreds, maybe a couple of thousand meanwhile. But if you now want to do an experiment, then you have so many tools already available. So it is possible for a single person to do essentially everything. And obviously you need, with these very high top instruments, you need staff that operate them. So it's seldom that a researcher goes to the microscope and performs the experiment. Rather, he gives staff the samples, and then they make sure that everything goes all right.
That's what the postdocs and the PhD students do, basically, right?
No, it's staff.
The technicians, really, you mean? Okay.
They are right. They manage the entire instrument. It is not very good to open such an expensive instrument to essentially everybody. Then you will have so much downtime that the whole thing doesn't work.
Yeah, if your instrument costs millions, then it's easy to pay someone
exactly
$50,000 a year or something to run it. Yeah.
Mm-hmm.
And those are beautiful machines indeed. Okay, well, thank you for saving lives, because basically what you're saying is that, like, indirectly through developing this technique, the Cryo-EM, it has now become like easier and faster to develop vaccines against novel pandemics, which ultimately saves hundreds of thousands of lives.
Right.
And helps me personally go out and have a beer in the pub, finally, which I'm so looking forward to.
I hope so. But right now it doesn't look like it, right? It's
no, I have to drink my beers at home. Yeah.
Right. Okay.
You're arguably the person in the world who can answer this question the best. What is cryo-electron microscope?
Okay, it's an electron microscope that is specially equipped to image a frozen specimen. Okay. So it leads to a question: what is an electron microscope? Obviously, an electron microscope works with electrons instead of light. It doesn't have glass lenses; it has magnetic lenses, and the electrons are produced somewhere, accelerated to high velocities by a anode and cathode and anode, and then they fly freely through vacuum.
It's a microscope that uses electrons instead of light. How does it look like? Is it big? Is it small?
It's very big. It's very big, and the reason why it's big is that it has very heavy machinery inside. The magnetic lenses are involve spools, windings of copper, and they have obviously iron, so they're pretty heavy.
Have you ever lifted one? Have you ever lifted one?
No, you cannot lift it. You can only lift it with a crane. It is pretty much the size of a room or even higher.
All right,
okay?
So it's not your high school microscope that you can bring in your backpack. And I assume they're also super expensive?
Yes, they're super expensive. And the high-end machines right now, they are being manufactured by Thermo Fisher, and they are something like in the order of $5.5 million.
Yeah. But you need the kind of equipment to make major breakthroughs and to make science that's actually gonna matter. But to come back a bit and just explain to somebody who has never heard of an electron microscope. So what do you use it for? You use it to check out very small stuff. And why is it important? Why is it important that you're looking at very small stuff?
Yeah, first of all, the difference between light microscope and electron microscope is that with electron microscope you can look at very, very tiny things. And light microscope is limited essentially to the wavelength of the light. So the wavelength is in the order of 400 nanometers. The wavelength, and the molecules are in the order of maybe 50 nanometers. Okay, they are so small that they cannot be seen by the light microscope. Now, why do we have to look at tiny objects? Well, the tiny objects that we are interested in biology are the molecules. Molecules are sort of the unity of life. The interactions between molecules, they sort of carry on life functions in the cell.
So by looking at that very small stuff, very small biological molecules, we can basically figure out life itself?
Yes. The life processes are constituted by interactions between molecules. That's what life is about, and not just in our body, but everywhere in the world. All organisms, including bacteria, yeast, plants, animals, all are essentially constructed from the same principle. Or on a molecular level, we are very similar to things like bacteria and so forth.
And that similarity is mirrored by the ribosome, right? So every single living thing on Earth has a ribosome, and the ribosome is at the core of its biological function. And you have been studying the ribosome, this very key molecule, arguably the most important biomolecule ever. What have you found out about the ribosome?
I first have to say that not all living things have ribosomes. Viruses don't have ribosomes.
Well, are they living?
Yeah, okay, that's a good question. Are they living? They're sort of parasites at the edge of life, I think, essentially.
So what did you find out about ribosomes? Maybe just in 2-3 words, what is a ribosome?
Well, the ribosome is a translation machinery. So there are certain molecules that are highly organized, and one calls them molecular machines. They're really sort of, you can think of them as machines that have a certain work cycle. They go around, they have a beginning and an end, and so forth. Okay, so one of those machines is the ribosome, and it's very complex. And what it does is it translates the genetic information into protein. So there are two languages in all organisms two languages. One language is the genetic code. The other language is the sequence of amino acids. These are completely different languages. One has to be translated into the other.
So basically,
and that's being done by the ribosome.
So the ribosome is basically an interpreter, right?
Yeah, it understands the genetic code and then manufactures a protein by putting amino acids together. And these languages are almost universal in the world.
You're talking about the ribosome and how it's the interpreter, and it's not just interpreting; it's also building our entire bodies.
Well, it doesn't just interpret; it actually builds the proteins. Okay, and it builds the proteins according to genetic instructions. And the proteins themselves are it's the stuff. These are the molecules that we have in our cells, and they are the ones that interact with one another and essentially constitute life functions.
Okay, so one thing I'm curious about. So the way I understand, you have received the Nobel Prize because you have made progresses in developing this tool that helps us understand ribosomes better. So you have basically made contributions to developing this very specific microscope that helps us better understand the ribosome. Is that correct?
Well, it doesn't just help us understand the ribosome, it helps us understand all molecules. I use the ribosome only as a demo, only to develop the tool. And I'm a physicist, okay? So I entered the entire field without knowing anything about biology. And then I developed this, I had this obsession of developing this kind of tool to look at individual molecules rather than crystalline molecules. Exactly. And I needed to have some kind of a demo, a molecule with which I could show that the whole thing worked. And the ribosome came along as a very convenient one because it was large, that was good. It was stable, which means that there were not too many changes between different ones. And it had high contrast because it has the so-called RNA that has a high phosphorus content. Okay. So these three, and then there's a fourth advantage of getting a ribosome. Almost nobody knew anything about the ribosome, about the structure of the ribosome. So, which means that if my method was going to work, then it would be a very huge progress. Okay.
And a huge progress.
So the ribosome served this whole purpose, and then in the end, the tool became available for thousands and thousands of other molecules as well.
And the kind of the breakthrough or the one insight that led to basically developing this kind of technique that sees down to the resolution of an atom. I mean, you cannot get a better resolution than one atom. Was, and correct me if I'm wrong, this insight about programming. So you were at the very beginning in the 70s, like doing computer stuff to try to get the resolution better. So it was not that you made the machine better. You basically built the brain of the machine that allow you to see way better than people could see with the same machine before. Is that correct?
No, it's even before that. It has something to do with the concept. There was right at the beginning when electron microscopes were first used for looking at molecules, people were in some kind of a mindset which was very rigid. They thought, and that's with the equivalence with X-ray crystallography, they thought structure only makes sense if you have crystal structure. Okay, there's no sense to even speak about structure if the molecules are not in a very regular arrangement. Okay, Does that make sense?
Yeah, sure.
Did you hear that?
Yeah,
okay.
sure.
All right. Yeah. So So people were just looking at at arrangements that were highly ordered, helical structures or icosahedral structures like viruses, or at planar structures, but always highly ordered. And and you can get structure out of this by making use of very fancy mathematical transformations with Fourier transforms, but it only works if the crystals are very highly ordered, and not if they are sort of ordered. Okay, and most crystals are not ideal, and so that imposed a restriction. And so the breakthrough was conceptual, to say, hey, why do we need crystals? What's so special about crystals? Can't we just use single particles, that are not clogged together? Yeah. So in the case of crystal, you have to tilt them in different ways in order to get all the view information. In the case of molecules that are in solution, they are already tilted. They are tilted themselves. They are all in random order, and they're randomly oriented, and you have thousands and millions of them. Okay, so it also means that when we are worried about radiation damage, molecules are burning up, and so you cannot expose molecules over and over again to the same beam. Exactly. And so if you instead spread it out over many copies of identical molecules, then you have the problem solved. You have three problems solved all at the same time by going to a separate molecule. And so this concept, was the crucial thing. And then you have to worry about programs. You have to say, what do I have to do in order to make it all work? Because there were no programs for this. Okay, so I had to make myself a workbench of programs. Many hundreds of programs that all work together in a modular way, so that I had something like a Baukasten to put blocks together.
Exactly. So there must have been a hard part too, because there was no Google back then to download code. There was no forums to check how to do that. You had to learn everything from textbooks and just trial and error. Coding in the 70s.
There were hardly any textbooks about this, really, either. But I had one great advantage. I was as a postdoc for 1 year, almost 1 year, at the Jet Propulsion Laboratory at Caltech, California, right? In California, and that was in 1971. At the time, they had the most advanced image processing in the world. And they had the kind of equipment, and they did already image processing of signals that came from space probes that were sent up. So it was it's a great environment.
That was image processing. You took a lot of pictures, or pictures of the same molecule but in different positions, and that's how you applied your knowledge from Jet Propulsion Laboratory to your problem in.
No. I didn't apply that knowledge. I simply was in an environment where they had a modular image processing system, and I used the infrastructure to dock my programs so that it became convenient to work with them. Right. So I had my own modules. I plugged them in, and I could take advantage of the entire programming infrastructure there.
And that led to the actual birth of what we call today cryo-electron microscopy. Is that correct?
No, that's not correct either, because all my work was really done with samples that were prepared in a very poor way. There was no Cryo-EM. There were no cryo methods available. At the time, the molecules were prepared by so-called negative staining. So that is done by putting a little heavy metal salt into the solution and then air drying the molecules on the grid. And when you do this, then a little puddle of stain forms around each molecule.
Okay.
And so what you see the molecule only because you don't see it. Okay. You see only the exclusion of stain and not the molecule itself. So it's a very poor method of getting information, three-dimensional information. And part of the problem is that this method leads to partial collapse of the molecule. But anyway, so I had this method to work with in order to develop my entire approach. And then when the cryo methods came along, I eagerly, used my programs on these new kind of samples, but the entire sort of the conceptual infrastructure and the math and the computation was already there to do that.
All right. So it was a combination of engineering and the insight. But you were the person to bring the insight along to say, okay, well, we do not need these crystallized molecules. We can actually do that with just random molecules, randomly aligned. Yeah. And that's what allowed you to see molecules in 3D. You did not see them at the atomic level from the beginning. That took you another 30 years, right?
Yeah, it took a very long time because your computers were not very big. The microscopes were not optimized. They had they didn't have very good illumination system. They were not powerful enough, and so forth. All this was a development, and when cryo methods came along, then electron microscopes became specialized. But the manufacturers always had to take a gamble because they didn't know how the field would develop. But these single particle kind of methods, there was not the only ones that was looked at with electron microscopes, because there were all these people that still looked at crystalline structures. But so the single particle methods was not the only motivation for manufacturers. Mm-hmm. For a very long time.
Talking about which, have you ever considered working for the electron microscope manufacturers, or was it clear for you that you were going to stay in academia?
Yeah. I couldn't think myself into an industrial environment. I really, it's part of it is only that I'm thinking. I'm used to doing my own thinking. Okay, I work very well in teams. But I need to concentrate, I need to figure out my own solutions.
Well, so we described the ribosome and why it's important. And so in the early 2000s, it first became possible to actually see ribosome, for example, down to the atomic level.
That was not, that didn't become available until the very fancy cameras came along. That was the big bottleneck that persisted until 2012.
Has that particular discovery, or has other discoveries, I'm sure there was like a lot of those, that led to something tangible? I think listeners might want to hear about a concrete story where maybe a medicine was developed. Like by understanding the ribosome of a bacterium, for example, you could target that ribosome or target that part of the bacterium to better kill it. So do you know of any such like concrete examples where something was discovered with this very precise electron microscope and it led to a drug, for example?
For the ribosomes, once you knew the atomic details, you were able to develop antibiotics. There are a lot of antibiotics, natural antibiotics already available, but now one could develop multiple antibiotics and very specifically targeted and so forth. Now, I have to say that along the way, as we improved resolution of the ribosome, then all of a sudden the X-ray structure became available. That was in the year 2000. And we were sort of left behind because they reached atomic resolution before we reached it. Okay? Now, there is an interesting relationship between the structures that you get out of X-ray crystallography and the ones that you get from single particle cryo-EM. In crystallography, the molecules are sort of pressed into a crystal, and they cannot wiggle around very much, so they are in a lowest energy conformation. And so this conformation that you get from X-ray crystallography is not particularly lifelike. Mm-hmm, yeah. Whereas when you look at them individually with cryo-EM, they can still wiggle around, and you can still account for all the different shapes and forms that they attain through their life cycle.
What's the temperature at which you're studying molecules in a cryo-EM?
Oh, this is the liquid nitrogen temperature.
Okay. So it's not like a crystalline mesh. It is like molecules are still kind of allowed to kind of like change conformation.
No, it's really the freezing is so fast that the molecule, in whatever the state they simply are solidified in and then surrounded by a glassy like.
Exactly, like an amorphous,
matrix. Yeah, it's an amorphous form of water, or a form of ice. And it's not a crystal form of ice.
Yeah. By the way, since we're talking about this, I'll ask you a question about cryopreservation. This is the technique to put people into liquid nitrogen to freeze people, well, basically to not freeze them, but to vitrify them. And so with the hope that we're going to be able to wake them up in 50 or 100 years and give them a chance to live longer. Have you looked into this? Have you heard of this? Are you interested in this?
I've heard of it. I'm not particularly interested in it, and I don't really know whether it's going to work.
Well, you don't know now, right? But so this is a question because I'm not sure how much longer you might live, but maybe you would want to see the future. How exciting would it be to wake up in 200 years and see the future? Doesn't that tempt you?
Probably be very frightening if it really were accomplished, you know. Yeah, I mean, you have a certain frame of mind, you have a certain culture, you're embedded in something, and then all of a sudden you're in a world where you need years of interpretation in order to even make sense of it.
Probably. Or maybe we are back in the Dark Ages, and maybe humanity has not managed to progress, but we have stagnated. Who knows?
Well, I wouldn't say that. Okay, I'm not equipped to answer your question about the frozen cadavers. Yeah.
Okay. So here's a question about the Nobel. Has the Nobel Prize changed your attitude towards research, and has it changed the way people treat you?
Well, I talk about the latter first. There's a noticeable gain in respect. I open my mouth and people don't interrupt me, and so I can speak freely. Nice. For me, it's always the handicap to see expectations of people, to see that they're waiting for me, and so I'm always worried about being interrupted. Okay, it's always been like this, and now I can speak freely, and this may make an enormous difference. That I can articulate myself without being harassed all the time. And there's also in groups there are always people who are pushing themselves forward, and they're sort of the bullies. And they are it no matter whether they have more experience or insight, they take their shots all the time. Okay,
yeah, that's what they do.
And that's what they do. So this kind of group dynamics, I've observed very, very much, because I sort of considered myself the victim there, and so all of a sudden the group dynamics is completely different now. So it's a huge difference for me, and it's also dangerous because people expect me to speak about things like you just before, about things that I have no experience about, that I have no expertise for.
Okay, so the other question was, the first one, if the Nobel Prize has changed your attitude towards research. Are you doing your research differently?
Well, it really means that I don't have to be so keen on achieving something. I can take risks and just think about this more as exploring things, even if they are high risk, even if I run into big problems. So it's not such a big deal anymore. and I'm one thing that also happened is that I'm not. I hardly will write review articles anymore because they are a pain. They are, review. You're bombarded with invitations to write review articles, and they're supposed to, bring you some more, recognition and glory and so forth. But it's very hard work, and obviously it doesn't pay anything, and so one has the feeling one is out of the rat race, right?
Yeah, it's good. It must feel good, like the part where people listen to you and don't interrupt you, and that you don't have to do the rat race part. What is intelligence? Is there such a thing as intelligence, or is it about just curiosity and stick-to-itiveness?
Well, it's really intelligence. Intelligence is really has been previously understood as something absolute and some kind of a scale, an IQ, but it's really specific. It's specific to social situations, to the environment in which you are. You can be intelligent in getting around in the woods, or you can be intelligent in the urban environment or in a particular skill. So it's really, I think people have different forms of intelligence, and if it could well be that they are not being challenged in their own specialty. Some people are unfortunate that they are in an environment in which they cannot really bring out their kind of intelligence.
Yeah, I agree, definitely. And I think this world is particularly dangerous for minds who can be captured by other stuff. You have the phone, you have video games, and I think it's never easy. It's never been easier than today to just get buried in that stuff, and that kills intelligence or whatever. What do you think about the nature versus nurture question? Talking about intelligence, or in general, all the stuff that we think as a combination of being nature and nurture. Where do you put your line? Is it more nature or is it more nurture?
It's much more nurture if you talk about individual differences. The nature part is for us as a species. We have evolved to have this kind of the capability of such mental sophistication, but it needs to be nurtured, and the very important part of the nurture is really the first few months. The first few months where all the
you think so? Okay.
where essentially all the networking grows, and the first real network forms.
Well, you could argue that the network continues to form until you die, right?
Yeah, sure, but but the challenges in which you're confronted with intelligence come early in life. You go into high school and college and so forth, and all these questions come up. Is he equipped? Is she equipped?
Yeah, exactly. If you had not acquired it when you were young, it's probably too late by college to start to get some more nurture.
Yeah. It is really it is more of an uphill struggle if the initial tracks are not laid.
So at this stage, I think I'd like to ask you the question about the smartest person ever. It could be a person who has died or who is still living. It's a weird question, right? With what's smart? You just described how it depends on how you define it. But who's the deepest and broadest thinker you have ever known?
I don't know. Plato, maybe. I mean, St. Augustine. You have to go through the entire range, I mean, there are people who are absolutely fabulous. Roger Penrose. There are people who have this immense capacity to look at very complex structures, and they can do math in their brain, and but they are very often very specialized. And to talk about someone who has such a universal.
Yeah, like Richard Feynman, who is not only good at his specialty, but he's also like all over the place.
Yes, he still is. He is. He has philosophical thoughts, but he will not really bring in that much on that. But you're talking about some kind of a universal mind, and they're really hard to find nowadays.
Oh yeah, for sure.
Because of the specialization of education and the specialization of the environment.
Exactly. It's another question if it's a good thing or a bad thing. It's out of our control. We cannot do anything about it. Specialization happens because that's useful, so to say. But let's see where it gets us. I think for technology, it's very important that we remain specialized. What about the weakest link in advancing research in your field today? Is this about a lack of brain power, or is it more about money or red tape? What's the weakest link in advancing technology today?
Well, money is a very big factor because there's really funding has been inadequate for quite a while in the United States, at least. I don't know really the situation in Europe, But all these things are sort of going up and down all the time. There's also there are also waves in there's sort of fads in the way science is funded, and one of the fads is for instance high throughput. If you do high throughput structure research, you just produce things and you don't know why you're doing it, and it becomes some kind of a heap of knowledge. And so to me, hypothesis-driven research is the way how it should be done.
And so it needs
Another
more money.
fad.
And so it needs more money.
Yeah, another fad is translational medicine. This is the idea, you heard about that, translational medicine.
Translational medicine?
Yeah,
what do you mean?
Well, the idea is really that there is a tight cooperation between essentially hospital, clinic, and research. Okay, so one thing feeds the other in some kind of a direct feedback manner, and it's a preposterous idea, because the usual development is that you have basic research, and then you have sort of that feeds the general literature, and then people pick it up and then draw conclusions and inferences and so on, and it builds up, and then maybe 10 years afterwards it really manifests itself in some kind of a medical advance. And instead funding for a very long stretch of time, it's still going on, went into fostering these immediate handshaking associations, between the bed and so forth. So even now there's the concept of personalized medicine. There it makes a lot of sense to have a direct feedback to find out what a person needs. One can, however, even that is based on years and years of basic research. Okay, this didn't come from nowhere.
So basically, you're saying that a big limiting factor is in this case red tape, because the administrative system is basically tying the hands of the researchers, and the researchers are kind of pushed to produce immediate returns, like money generating discoveries.
And they're not capable of doing that because it doesn't work. So they have to essentially rationalize what they're doing. They have to sort of make up grant proposals that conform with these kinds of desired interactions. Yeah. And you see this everywhere, and so my point is that it's not just lack of money. It is also the wrong kind of emphasis, the wrong kind of incentives.
Yeah, it's the incentive of big old cash, and I think it's kind of sad that cash plays such a big role in fundamental research these days. Do you have any info on whether it's better or worse in Germany versus the US? It's probably the same, right?
I don't know. It's very complicated in Germany because there's this European funding, and I don't know to what extent that has a very big role. There are sort of different levels of funding. So I really lost track of this.
I'd like to talk to you about existential threats. I'd like this world to continue on developing new cures and being a happy world for another million years. But I think there are some existential level risks that could end this human project. Do you fear that something could go very wrong and kind of terminate our civilization?
Yes, yes.
What's your top candidate, the top three candidates for such a scenario?
Well, the global warming is a good candidate. It's almost the only one that I fear. Uh, Obviously, we can also deplete the resources, even if we are in a regular climate. We can deplete the resources by simply overpopulating, and then in the end, we wind up with incredible levels of calamity. We wind up in wars and so forth.
So I really find it interesting that you're talking about global warming and overpopulation. Those to me are not civilization level threats. In fact, I think it's a non-threat. Those are both like very slow processes, and they're gonna find some natural equilibrium, to my mind. When I think about threats, I usually think along the lines of a nuclear war or an AI explosion, a singularity, or like a pandemic. What do you think about those?
Yeah, sure. Yeah, okay. Nuclear war. Nuclear war. Obviously, we have been living under that threat, and seeing the instability of a person like Trump makes me really worried that it can happen at one time, and maybe because he has an extended family, that's the only thing that saved us here. The other one is the pandemics. As a threat to civilization, I don't see it as a threat to civilization. I don't see it as a threat to survival of the human species.
Wow, it's interesting, right? Because these very small molecules so well, and they could be engineered. Like these days, it costs $100 to edit genes by the letter. Don't you worry that this could be so easy and so accessible that somebody could like cook up the next virus?
No, I'm not worried about viruses because of the selection process that takes place. They are always survivors, and these survivors then, essentially create a new species, and so there will always be people left. This has always been the case, and so I mean, obviously, civilizations get destroyed in the process. However, it's not that the human species cannot survive.
Yeah, the same thing as with maybe overpopulation or climate change. I don't think that it would like finish our project. It just would create a big major hurdle, but I think we would definitely survive that one too. What about artificial intelligence and super intelligence?
Well, I'm essentially, I'm very worried about this, and there are the two aspects of this is that intelligence could be could actually be could come to a level where it becomes a threat. I mean, it's like in the scientific sci-fi movies, this kind of threat, and which I never took serious. But now you have even self-replicating apparatuses. They have been machined as a demo. That can be done, but then the other threat is really the way we become accustomed to it, and the way it really makes profound changes to our culture. And then the other one is really the use of AI in governments, like in China, where you have universal as a universal tool. You have face recognition. You have yes, and you have essentially AI data mining of the entire social. I mean, the entire social fabric.
Exactly, and the question is, yeah, and what is the Chinese Communist Party gonna optimize its function for? Like what is the AI gonna optimize for? It's probably gonna optimize not for the well-being of the people, but probably for the survival of the Communist Party, which I think are pretty orthogonal concepts.
Yeah. But they have a certain, the Communist Party or the leadership has a certain concept of what the well-being of people is, and that's what they optimize it for, and that's
unfortunately,
yeah. So it's well, one can talk about ages about this. It's
well, feel free. I have a lot of questions that I can throw out. So, for example. Consciousness. You have looked at single atoms. Like there's no atom that you could not see today, in principle, if you looked hard, which means that we could, if we try hard, we could basically map the entire human brain, the entire human body to the atom. Do you think that consciousness is something undiscoverable? Is it something ethereal that we cannot understand? Is there like some limit to how much we can understand, or can we understand consciousness?
Well, it's very hard to understand how consciousness is constituted. One can see how all the functionality can be generated by neural networks and so forth, but one cannot understand where the insight view comes from. The awareness. Where does the awareness come from?
It's unknowable.
No. I'm not an expert in this. I wouldn't simply say it's unknowable. It's simply for me hard to conceive how one can get a clue of this.
Okay. I am of the camp who supports the idea that consciousness is knowable. I think consciousness is probably something very simple in a way. And when we discover what it is, which is going to be like this and this and this part of your brain, then we're going to be like, okay. So I don't see anything special about consciousness apart from the fact that this is my reality.
Well, maybe you have some insights that I don't have.
I don't have any insights. I'm just a pure materialist, and so are you a pure materialist?
Oh yeah, sure. I'm only saying that it's hard for me to figure out whether our intellect is sufficient to get behind it, to get to these explanations.
That's another thing.
I'm not saying it's unknowable. I would only say it's unknowable with these kinds of restrictions, knowing our own limitations. Now, I know somebody very well who is pondering these questions. His name is Wolf Singer, and you might have heard about him in Germany. He is a good friend of mine. Okay, so
I should talk to him probably. So just one thing more about consciousness. So, materialism. If you are a materialist, you said you consider yourself a materialist. Do you think that there is such a thing as God?
No.
Okay. What about this next one? Is it possible that when we discover something like the great unified theory or quantum gravity, some really profound equation about our universe. Do you think that could reveal something about the interface on which this is running? Because to me, it's pretty clear that the universe is so mathematical that it's a processing. It almost feels like a program running on a computer. And I would like to know if you feel the same. But I also would like to ask if you think that if we figure out how exactly that program works, our reality, could it give us an insight to something like God?
I mean, God is just a label.
Exactly.
And
Exactly.
and it's a label for the unexplainable, and then I mean people are sort of clueless in many things, and there's some kind of a concept that is applied, and he's imbued with certain qualities and so forth, and so you could this entire spectre that you see when you get these insights as a physicist of the biology in the tiniest details, then you wonder, how could it all happen? How could it all come? And
emergence.
People point at evolution, but how come that we have laws in the universe that allow this to happen? And so you can always ponder like this, and you say, that this entire insight, this awe experience is somehow, you say that's God. Okay, there's something behind it.
Yeah, define God. I mean, God is this guy who's running our big reality program. This is how I think of God. Is the programmer who has clicked start on the universe.
Yeah, I mean, you have right at the beginning the Big Bang, and then there's this plasma, and then all of a sudden you have the differentiation, and then you get some elements, the formation of these elements, and all these elements are then on the way to become building blocks of life.
Yeah, it's really beautiful. We are all remnants of a long-gone supernovae, right? Speaking of which, what do you think about space exploration? I'm a big fan of space exploration, and I have worked to develop ways to reduce Martian soils into oxygen and metals to settle these planets and use materials that we can find there. Do you think space exploration is a good endeavor?
I don't know. It seems like if we run down this planet in the way we've been used to, then it might be a good escape. But I find it as a concept just very dismal. I mean, I find it dreary to go into places that are so incredibly hostile, and I mean, I guess I admire people who put their life into it, but there's nothing else that
I really like that there is a country over there, over the pond, the U.S., and I think the kind of people who want to go to Mars today are the kind of people who wanted to go to the U.S. 400 years ago. And I think it was a really good idea that they colonized the very hostile, very, very uninhabitable U.S. soil. And
well, it's all within this planet.
But it was incredibly hard to do back then.
Yeah. Well, it's not a... I mean, it's a comparison of scales that are incredible. The scale is completely different. You're scaling it up in such a way. We're talking about millions of dollars,
billions,
billions,
if not trillions. Yeah, it's a very actually, I did run some numbers on this, and actually, it would be possible to colonize Mars on something like 5% of the U.S. budget. And obviously the U.S. is spending something like 20% of its budget on war. And so what's better, war or colonizing? But, let's put the subject behind. I'm a big evangelist for space exploration. Could we do the German questions now? I'd like to talk some German with you.
Yeah, okay.
What's your secret efficiency booster? Is it diet or psychoactives or exercise? Like, do you have like a way to ensure that you function at 100% efficiency in your brain?
I'm not 100% anymore. So it's reduced, and I have coffee, and what really keeps me going is the ability to switch between completely different activities. And one of them is writing, fiction writing. I'm writing fiction. I was
going to ask about that. Could you tell me about that? Why do you write, and since when?
Well, that's for ages. I already did some in German, but then when I came to the United States, I started writing in English, and I got a lot of encouragement. I went into writing workshops with. Some of the teachers are really renowned writers, and so I got a lot of encouragement from them. And then I started writing short stories, poems. I just published a novel, which is in my signature there. It's called An See.
Okay.
And I have 4 other novels that are unpublished. They're sort of on the back burner. So I'm writing constantly, and it's really very inspiring to me to create worlds.
I love to do that too. I like to write, and I also like different types of writing. It's just a creative exploration. A role model. Do you have a role model today, or did you have a role model before?
I didn't. I always looked up at Nobel laureates. I thought that this was an unbelievable achievement somewhere, and I didn't know how they got there.
Now you know.
And so yeah, right. Through hard work, but no, there was no particular role model. I can't.
One person.
Never. Okay, I have to rush off. I was called to dinner.
Oh,
right?
Sorry for taking so long. All right, so
take care.
fun. Was a lot of fun.
Okay,
Bye.
bye.